Hi everyone, my name is Andrea Salzinger. I'm a PhD student at the UK Dementia Research Institute at the University of Edinburgh in the lab of Dr. Bhuvanesh Selvaraj and Professor Siddharthan Chandran. And today I'm going to tell you a little bit about my project, what I have been doing over the last 4 years. As you can see, the title of my presentation is Using Human Stem Cells to Model Neuromuscular Junction Degeneration in Motor Neuron Disease. And that is what it captures in a nutshell. But I'm gonna walk you through the more details now in the next few slides. First of all, what is a neuromuscular junction? As I'm gonna mention it a lot, the acronym is NMJ. But what does it do? It's very important to transmit signals from the brain to the skeletal muscle to allow for several functions of the body. For example, to allow that you're able to move yourself using your mobility, to look left and right, moving your eyes up and down, to be able to speak what I'm doing just now, or also to be able to run, because basically it controls all your movement and everything that you think you want to move, it allows you to then execute this movement. But what happens in a person that lives with motor neuron disease? What you can see in this figure is the spinal cord, a section of a spinal cord, so only a part. And then in blue, you can see a motor neuron nerve that is actually innervating the skeletal muscles, which you can see over here. And all of these tiny connections, those are the neuromuscular junction. And what you can see over here is the circuit that a healthy person would have. But what happens in motor neuron disease is that the axons retract back towards the motor neuron, which ultimately leads to motor neuron death. Now, in my PhD, what I wanted to understand are several different questions. One, why does the motor neuron and the neuromuscular junctions degenerate in motor neuron disease? And is there anything that we can understand by studying those neuromuscular junctions that can allow us to reverse the degeneration, prevent it, or promote compensatory mechanisms, for example, that one axon innervates another skeletal muscle fiber, as you can see here with this green and blue axons. In order to study this, we use pluripotent stem cells and in specific human pluripotent stem cells. And that is important because motor neuron disease is a human disease. You can't just have a mouse that randomly develops motor neuron disease. And in order to do so, pluripotent stem cells allow us to model the disease in a very specific way for humans. So how this works is we take blood cells or skin cells from any different person that we could have, then convert them into pluripotent stem cells via a very well-established protocol. And then with those stem cells, you can again model different cell types of the brain, but also of any other part of the human body. But given that I'm interested in brain cells, for example, you can make astrocytes, oligodendrocytes, which are helper cells of the brain. And more importantly for me, you can make neurons. Now, how will we take this a step further to now model the neuromuscular junction, which is a connection between two different cell types, the motor neuron and the skeletal muscle. So in order to do that, we are using motor neurons from stem cells, but this time in a 3D structure, and then fuse them together with a 3D structure of skeletal muscle cells, which then will give us a so-called neuromuscular assembloid. And you can see that the motor neuron reaches out to the skeletal muscle and forms this neuromuscular junction. So how does this look like in real life? What you can see over here is a neuromuscular assembloid, which has been cut very thinly to allow for the proteins to be visualized via fluorescent tags. So what this means is under the microscope, we can look for certain proteins. In this case, I was looking for protein that stains the nerves, which you can see in blue, over here, here, and over here. And then I was looking for proteins that stain the muscle fibers, which you can see in yellow. And the skeletal muscle sphere is surrounded by the dotted white line. So basically what we can see here is that the motor neuron and the skeletal muscle fibers were actually interacting with each other. But then we were obviously also interested in seeing, do they also form these NMJs? And what we can see over here, by just zooming into the tissue, is that they indeed form neuromuscular junctions. So in white, you can see the muscle fiber. In green, you can see the nerve, so the motor neuron nerve. And in red, you can see the neuromuscular junction, which shows us that they have been formed. Taking this a step further, we were also interested if these neuromuscular junctions are functional, which means when the brain sends a signal, are the muscles actually able to contract like we would? And what you can see in this video is that indeed the skeletal muscle fibers start to spontaneously contract, in this case 2 months post-assembly, so meaning 2-month-old assembloids. But we have already seen very early contractions of muscle fibers soon after we have made the assembloid. And this is importantly only happens if we have motor neuron and skeletal muscle together. So overall, this has shown us that we are able to model the neuromuscular junction in a dish in a 3D environment, and that it generates functional neuromuscular junctions. Now, taking this a step further, we wanted to apply it to motor neuron disease. And in specific, I was interested in the protein called TDP43. So in a healthy neuron, TDP43 is expressed in the nucleus of a cell, as you can see here in the green dot. But what happens in an MND neuron is that initially TDP43 is expressed in the nucleus, but then it gets taken out of the nucleus into the extranuclear space, into these little aggregates. So it basically forms clumps. And these clumps, or the loss of the nuclear TDP, are then leading to the degeneration of the neuron. So my question was, what happens if we have motor neurons without TDP43? Can they induce the degeneration of the neuromuscular junction? And for that, I took healthy motor neurons and fused them with healthy skeletal muscle. On the other hand, and I made a healthy assembloid. And on the other hand, I made a healthy motor neuron, I made a motor neuron without TDP43 and fused it with a healthy skeletal muscle, and then made an MND assembloid. And what we were observing was then looking again at the neuromuscular junctions, is that indeed we see a decrease in innervated neuromuscular junctions in the MND assembloid, which basically recapitulates what we see in the patient. And that was a very novel finding because no one has shown this in a human system before, that TDP43 loss alone can induce the neuromuscular junction degeneration. And that suggests that TDP43 has very, very important functions within this nucleus of a cell. And if we lose it, that could actually start the disease. Now, one option of trying to reverse or prevent this neuromuscular junction degeneration was by enhancing the energy that is delivered at the neuromuscular junction, because there have been several different research approaches which showed that in MND, the energy delivery fails often. So what we did in this case is we used, again, the healthy assembloids, which had around 40–50% of NMJs, as I've shown you before, an MND assembloid, again without TDP43, and then we used an MND assembloid, but in this case, we improved the energy delivery to be enhanced. And interestingly, we were seeing that it indeed prevents the NMJ degeneration. And with that, thanks everyone for listening. Thanks to the lab that has supported me a lot. And obviously thanks to all the funders. Thank you.